The overall goal of this procedure is to visualize and quantify changes to biofilms in response to sputum and antibiotics.
This method can be used by researchers to understand key questions such as how the efficacy of various antibiotics against bacterial biofilms can be impacted by the presence of sputum. The main advantage of this methodology is it allows individuals to see changes occurring in biofilms in response to different factors.
To begin the experiment, record the volume of the previously obtained sputum sample. Add phosphate buffered saline, or PBS, at 2 times the volume of the sample. Next, use a transfer pipette to thoroughly mix the sample. Vortex the sample on the highest setting for 1 minute to mix it completely. Aliquot 1 milliliter of the mixture into 1.5 milliliter microcentrifuge tubes. Then, spin down the tubes. Following centrifugation, save the supernatant and discard the pellet. Filter sterilize the supernatant through a 0.22 micron filter and collect it in a clean microcentrifuge tube. Store the sputum supernatant at -80 degrees Celsius for future use. Place 40 microliters of previously prepared culture into 4 milliliters of fresh luria broth, or LB media, and grow the bacteria to obtain a culture with an optical density of 0.55 at 600 nanometers. Next, dilute 100 microliters of 0.5 OD culture into 400 microliters of prepared 10% sputum.